Good morning everyone. Today's topic is ear, concerned with hearing. Okay. Now, ear is the organ of hearing. It is divided into external ear, middle ear, internal ear. Coming to external ear, the external ear has two parts, pinna and external auditory meatus. Coming to the function of external ear, the external ear collects the sound waves and conducts it to the tympanic membrane. You can see the structure of ear here. From tympanic membrane, the green one in the middle, to the pinna auricle, it is the external ear. The pink one is from the tympanic membrane, the green one, and the pink one is the middle ear. Then the violet one is cochlea, and you can see cochlea. From cochlear nerve comes now. So cochlea has two parts, labyrinth and cochlea. You can see in this picture, in the middle ear you have malleus, three ossicles, three small bones called ossicles, ear ossicles. They are malleus, incus and stapes. In the pink part you can see white bones. They are malleus, incus and stapes. And green one is tympanic membrane. Then you can see external auditory canal and auricle. These constitute the external ear. In the middle ear you can see the ear ossicles and eustachian tube. Then in the cochlea, the violet one is the purple one is the cochlea, and you can see three semicircular canals. Utricle and sacral are not very clear. Okay, now middle ear contains three small bones called ossicles. They are malleus, incus and stapes. The middle ear also contains two small muscles called tensor tympani and stapedius. The medial side of the middle ear contains a bony wall with two openings. The superior opening, that is the above one, is called oval window, and the inferior one, that is which is below the oval window, is the inferior opening is called round window. In the picture, round window is shown. You can see again. Now, oval window, the foot plate of stapes is attached to the oval window. Round window is closed by secondary tympanic membrane. The anterior wall has two canals. The upper one contains tensor tympani muscle and the lower one is called eustachian tube. You can see eustachian tube in the picture, which connects the middle ear with the oral cavity. Now, functions. of the middle ear. The muscles of the middle ear conduct the sound from external ear to the internal ear. They also amplify the sound. They provide impedance matching for the sound waves in the air and movement of the base of the stapes in the cochlear fluid. The small muscles, tensor tympani and stapedius, help in protecting the auditory receptors, that is receptors for hearing, against loud sound. It is called tympanic reflex. The eustachian tube helps to equalize the pressure on either side of tympanic membrane. Now, tympanic reflex. It is a protective reflex which prevents strong sound waves from causing excessive stimulation of auditory receptors, resulting in their damage. So, it prevents damage of the hair cells, auditory receptors. The loud sound initiates reflex contraction of tensor tympani, which pulls the malleus inwards, and stapedius muscles, which pulls the foot of stapes outwards. This decreases the sound transmission. The reaction time for this reflex is 40 to 60 milliseconds. The tympanic reflex fails to protect the auditory receptors from gunshot sound, which has shorter reaction time. So, for the reaction time, that is, what is reaction time? From the stimulus to the response, that is reaction time. That is 40 seconds, 40 to 60 seconds, whereas gunshot sound has reaction time less than 40 to 60 milliseconds. So, it cannot protect the ear from loud damage. Coming to impedance matching. The ossicles, that is small bones in the ear, malleus, incus and stapes, they amplify the sound during conduction of sound. The arrangement of ossicles increases the sound pressure by about 1.3 times the difference in the size of tympanic membrane, and the 1.3 times, and the difference in size of tympanic membrane and the base of stapes increases sound by about 17 times. 17 multiplied by 1.3 is 22. The effective increase in sound pressure is 22 times. Here a lot of physics is involved, so no need to know the basics of physics, but here what you have to remember is the ossicles are arranged in such a way that they increase the sound pressure by 1.3 times, and the area of tympanic membrane and the base of stapes is, the difference is 17 times. So multiply 17 times into 1.3, it gives 22. So effective increase in sound pressure from the tympanic membrane to the stapes is oval window is 22 times. This amplification is required to overcome the resistance offered by the fluid in the internal ear. Internal ear has fluid, so the sound internal ear for movement of base of stapes. This is called impedance matching. When you produce sound, vibrations are set up in the air. These vibrations, when they hit the tympanic membrane, those vibrations are carried via the ossicles to the oval window. In the oval window, just adjacent to the oval window, there is internal ear. So in the internal ear there is fluid, perilymph and endolymph. So when you make a sound, it is transmitted to the perilymph and endolymph. When the perilymph and endolymph receive these waves or sound waves, their basilar membrane is set up into motion. Okay, and it will cause help to hear. Now inner ear. The inner ear is made up of two parts: bony labyrinth, membranous labyrinth. Bony labyrinth is a series of channels in the petrous part of temporal bone. Inside these channels is the membranous labyrinth. The fluid surrounding it is called perilymph. The membranous labyrinth is filled with endolymph. The perilymph resembles a CSF and endolymph resembles ICF. What is ICF? Intercellular fluid. No. The inner ear has vestibular apparatus, cochlea. Cochlea is a system of coil tubes 35 millimeters long, and it makes 2.5 turns, 3/4 of a turn. Now cochlea consists of 3 tubes lying side by side. Scala vestibuli. The 3 tubes are scala vestibuli, scala media and scala tympani. You can see in this picture. It is cross section of the cochlea. It is snail shaped. Cochlea, to see, it is a snail shaped organ in which when you cut the in cut section you can see 3 tubes, that is scala vestibuli, the above one, blue one, and the next one below it is scala media, and below scala media is scala tympani. So between the scala vestibuli and scala media, the membrane separating it is called Reissner's membrane. You can see in the picture Reissner's membrane. Between the scala media and scala tympani is the basilar membrane. Down it is written basilar membrane. On the basilar membrane, the hair cells are or organ of corti is present. Now, scala vestibuli and scala tympani contain perilymph. They communicate with each other at the apex of cochlea through a small opening called helicotrema. Scala media contains endolymph. Scala vestibuli has an oval window which is closed by footplate of stapes. That is, oval window is closed by footplate of stapes, and adjacent to oval window is a scala vestibuli. Now, scala tympani ends at the round window. It is covered by secondary tympanic membrane. Scala vestibuli and media are separated by Reissner's membrane. The first one, upper one scala vestibuli, middle one scala media are separated by Reissner's membrane, and scala media and scala tympani are separated by basilar membrane. You go back to that picture and see once again. On the surface of basilar membrane lies the organ of corti containing hair cells. Coming to organ of corti. Organ of corti is a receptor organ that generates the nerve impulses in response to vibration of basilar membrane. The receptors of the organ of corti are hair cells. They are of 2 types. Hair cells are of 2 types. External hair cells or outer hair cells, and internal hair cells or inner hair cells. The hair cells are covered by thin viscous elastic membrane called tectorial membrane. In this picture you can see organ of corti. Organ of corti is present on basilar membrane. Basilar membrane separates scala media from scala tympani. You can see basilar membrane on the right side. It is a basilar membrane and 3 rows of outer hair cells. Then tunnel of corti. Tunnel. You can see one tunnel. It is, and 2 rods on either side are rods of corti, and next to the tunnel you can see inner hair cells, and tectorial membrane you can see. Then stria vascularis is present in the bone, where they will secrete the endolymph, and you can see Reissner's membrane, scala media, scala vestibuli, and from the hair cells, pink one, you can see black fiber is coming. It is called cochlear nerve fiber. Okay, now we will go to organ of corti. The base and sides of the hair cells synapse with a network of nerve endings of the cochlea. The nerve fibers from these endings lead to spiral ganglion of corti, which lies in the modulus of cochlea. The spiral ganglion of corti in turn sends axon to the cochlear nerve. Coming to hair cells, they are embedded in the epithelium made up of supporting cells. The basal part is in contact with afferent neurons. The apical part contains rod-shaped processes called hairs. Excuse me. హలో చూసినా నేను క్లాస్లో ఉన్నాను 10 minutes తర్వాత కాల్ చేస్తాను వాట్సాప్ కాల్ చేస్తాను అది ఆష్మికి ఎగ్జామ్ అవుతుంది లేదు ఇది అమ్మ ఫోను ఆష్మి దగ్గర ఉంది నేను క్లాస్లో ఉన్నా నేను మాట్లాడతాను The hair cells are embedded in the epithelium made up of supporting cells. The basal part is in contact with the afferent neurons. The apical part contains rod-shaped process called hairs or stereocilia. Hairs are covered by thin, viscous but elastic tectorial membrane. The resting membrane potential of the hair cell is minus 60 volts. Coming to applied physiology, deafness. Deafness is inability to appreciate sounds. If deafness is present since birth, the individual is unable to speak. Hence, the ability to produce sound, for the ability to produce sound, the person has to hear the sound. To have, that is, it's not to have the ability to produce sound, the person has to hear the sound. Types of deafness. There are 3 types of deafness: conductive deafness, sensory neural deafness, and mixed deafness. Conductive deafness, if there is some wax or otosclerosis or anything which obstructs the passage from the external ear to the inner ear, it is called conductive deafness. And any damage to the hair cells or the cochlear nerve till the brain in the pathway, auditory pathway, it leads to sensory neural deafness. And if both are present, it is called mixed deafness. Now, how do you test for hearing? Hearing test, there are 5 types of hearing tests: watch test, Rennie's test, Weber's test, absolute bone conduction test, and audiometry. Watch test, you keep watch near the ear of the person and ask if he is able to hear the sound. If he is able to hear the sound, he is not having any deafness. But if he is unable to hear the tick 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 sound of the watch, he is having hearing defect or deafness. So Rennie's test and Weber's test and absolute bone conduction test, these are done with tuning fork. And audiometry, they are done in hospital where with audiometer, computerized audiometry is done. Pure tone audiometry. Okay. This finishes the class. Thank you.